Looking at this data, you can see that osteoclastin is a hormone and it's inactive initially. Osteoclastin is then activated later on by the pH decreasing. So the first question says, how is this an example of positive feedback? So in positive feedback, when something happens, it then causes more of the more of a change. So in this example here, we can see that when osteoclastin is produced, it interacts with the B cell, but that then causes more insulin to be released, and then more insulin release interacts with the osteoblast, causing more osteoclastin to be released. So it's the fact that osteoclastin causes more insulin to be released, and then more insulin causes more osteoclastin to be released. Wow, that's an example of positive feedback. For this question, it looks quite a straightforward question, but a big mistake that people made was thinking that osteoclastin is an enzyme and referring to active sites. It's quite a sloppy answer in a lot of cases, so there's a lot of reject points which I couldn't give if you mentioned active sites. So, changing pH, acidic pH, what's it going to do? It's going to break hydrogen bonds or ionic bonds. What does that do? It changes the tertiary structure, and that'll be both marks. It's only if it's an enzyme you talk about changing shape of the active site. Similarly, if it was talking about antibodies, you could talk about a change in the shape of the antigen binding site. You have to make sure you know what type of protein you're talking about. This question is talking about how respiration can be increased when insulin binds to a cell, and how respiration can be increased inside these cells. It doesn't state aerobic respiration, so it's not to do with oxygen specifically, and insulin doesn't really have anything to do with oxygen anyway. So it's got to be to do with glucose. How can the glucose concentration inside the cell increase? Because glucose is a substrate, it's going to increase the rate of those enzyme catalyzed reactions in respiration. So how does that happen? Well, when insulin, using your knowledge, when insulin binds to a cell, it causes more channel proteins to be inserted in the membrane, so more glucose will enter the cell by facilitated diffusion. Say with the marks. So it's understanding the idea that if you're increasing glucose concentration inside the cell, there's more substrate, so there's more enzyme reactions for respiration. For this question here, hopefully you can see that there is a correlation here, and that leads you to the name of the test. So the name of the test would be a correlation coefficient, or you could say Spearman rank. And then why would that be the case? Because you're looking to see if there is a relationship or a correlation between two variables, or you could say between the amount of CD20 on the surface and the percentage of B cells destroyed. For question 2.2, it asks, what can you conclude about the effectiveness of the drug rituximab on treating patients? So hopefully you'd start off with it's a yes and no. So what's the agreed point? And the agreed point is the idea that more of CD20 on the surface, the more B cells are destroyed. But there's quite a few negative points. First of all, you don't get any with 100%. So in no situation is 100% of the B cells killed. So it only destroys a maximum of about 80%. Also, we don't know if these B cells are cancer cells, so we don't know the proportion or the percentage of cancer cells that are killed. Because it doesn't kill 100%, it's not going to cure the cancer, but it will slow down the growth. So it's that idea that there'll still be some cells remaining, some cancer cells remaining, which could still uncontrollably divide. Another thing to bear in mind, negative point, is below about 5 CD 20 on the surface of the cell, then there's very little effect. This question was fairly well answered. The key to this is working out that it's asking you per decimeter cubed, and it's just wanting a straightforward difference. So difference between the number of B cells per decimeter cubed. First thing you have to do is work out the difference of what is the number of B cells per decimeter cubed in each, and it tells you here that you've got 1 times 10 to the 9 B cells per decimeter cubed. So each of these numbers you would divide by 10 to the 9, and then you just take them away from each other, and that's the difference. So the answer for this take away the answer for that. The key is looking at the units, it's per decimeter cubed, so each of these numbers is divided by the number of B cells. Okay, looking at this question, it does say use all of the information, and it's talking about a mutation of notch 1. How can it lead to the difference of B cells being destroyed by rituximab? Most of you just went for the final mark point, that the idea is not enough CD20 with the mutation, so you've got nothing for rituximab to bind to on the surface of a cell, so the B cells can't be destroyed. But you missed the points about suggesting how the mutation can lead to this. So first of all, what is a mutation? Due to the protein, it's going to change the tertiary structure of the protein, of the transcription factor. So therefore, the transcription factor can't bind to the promoter on the CD20 gene. So therefore, there's going to be less CD20 produced. And that was the key 3 points that you needed to say before you went to that final point, which most of you started off with. How the mutation can cause it. Another common thing that people didn't say is you need to say that it's CD20 that is being affected. So you can't just say a transcription factor can't bind to the promoter of what. You've got to say to the promoter of the CD20 gene. So be specific.